Good morning, everyone. So I'm Kevin, and from the Department of Radiology and the Senior Research Scientist. So my research topic today is development positron emission tomography image agent for the brain lymphatic system. So in the first few slides, I give you an overview of what we are doing or what's our whole program. Then I give an update regarding the project supported by the INBRE pilot. So our research mainly focus on molecular imaging. So what's a molecular imaging? So molecular imaging is a type of medical imaging provide what's happening inside the human body at a molecular or cellular level. It can provide quantification, visualization, and the characterization of the living biological process in our human body. So here show the different modality of the molecular imaging. Probably everyone heard about the MRI, the magnetic resonance imaging, ultrasound, or CT, and quite sure from the clinical side, that's the most open clinical daily practice used imaging modality. So, and each modality have the different characteristics, advantages, and limitations. So most of the imaging modality can be translated to human research or practice, particularly MRI or ultrasound CT, very long, and also less clinical use of optical imaging. There's we call the limited penetration or quantity yield, so less used. So today we're focused on PET imaging. So write 3 for the PET imaging and spectral imaging for the preclinical research. So what's a PET imaging? So this cartoon shows the PET imaging. So PET imaging uses a radioactive radioisotope to aid diagnosis, to monitor disease progression and respond to treatment. So the positive charged electron, they call the positrons. So positrons travel a short distance and annihilate with the electron, because electron negative charge, and they annihilate with the positive charge the electrons to release 2 photons. So from the picture, you can see the gamma ray. The high energy photons travel a short distance and on opposite direction. So the energy of the photons, they're detected by the multiple detector around the patient. So imaging was reconstructed by the computer software to provide the origin of photons by the back projection method. So typically there's a ring there, so they did around detector on the patient detect the origin where the radiation exposure, where the photons to look at according to the radiation extent, to look at the concentration accumulate in the disease or normal organ tissues. That's the basic rationale of the PET. So this cartoon shows the rational design of PET radio tracer. So right side, so just to give you the example, so we are trying to look at the tumor or disease, they typically overexpress some biomarkers, just like the red sign. So the left side, the general design of PET ray tracer, they typically consist of 3 parts. So right side, the green part is the target molecular. The typically target molecular have high binding affinity and selectivity toward the target. And the left side, the radioactive sign, probably everybody knows about the sign regarding sign, the radiation exposure. So that's for the signal detection. That's when we inject to animal human bodies. So this part, that's a signal, emit a signal. We use this radioisotope to detect the photons, the X-rays, emit from the molecular compound. So then we design a linker, try to connect the radioactive part and the green part, the target part. So the whole part consists of the radiopharmaceutical. Sometimes we also call the radio tracers, because when we inject to animal or human, one concept we need to keep in mind is the mass is very minimal. When we inject the dose, the typical mass is less than 10 microgram. That's 10 microgram to 100 microgram. So the that's why we also call radio tracers, because they will cause minimal pharmacological effect. They only maybe have sometimes high dose, maybe they only have radiation exposure concerns. But in terms of the toxicity study or regarding the mass, that's very minimal. So that's why the radiopharmaceutical we sometimes also call the radio tracers. So our research is made that the diagram shows our general research strategy. So we always started from the clinical challenges. So we have a lot of collaboration with the clinicians, physicians, we like to identify the clinical challenges. So because we are working in the children's hospital, so we work with the pediatrics and also radiologist, a neurologist, and oncologist to identify clinical challenges, what's the research gap. Then the next step, we try to identify a target we are interested in. So what's the target? And the clinical side, there's some challenging, maybe let's say some subtype selectivity, and also very challenging use a traditional MRI or CT, or sometimes very challenging to differentiate the benign tumor or for a malignant tumor. Just to give you an example, then just to identify a target we are interested in. The next step, we design new probe and with high binding affinity and selectivity toward the target. Then we develop a method for the radio labeling, just like I mentioned in the cartoon, how we incorporate radioisotope to the molecular. That's a key part. But because when we have good drug with the in vivo behavior, so how we monitor the drug, the ADME metabolite absorption and also distribution. So that's why we introduce something like a tag to the parent compound. But when we introduce the tag, we need to make sure that the new tag will not affect the binding affinity, sometimes even increase the binding affinity or modulate the water solubility. So that's why we need to introduce the tag, then we evaluate the property to make sure the minimum change of chemical behavior. Then we also need to test the stability ex vivo, in vitro. So that do all the stability test, in vitro biological assay to look at the binding affinity stability test. If all the in vitro behavior show that promising, then we move to in vivo animal studies. That include, let's say, biodistribution studies to demonstrate the binding specificity. We do some blocking studies and also do some dynamic PET imaging, a static imaging. If from the in vivo animal behavior still very promising, then sometimes we move to higher species. Before I joined here, I work in WashU. We have around, I think, 8 to 10 monkeys in WashU. So I involve a lot of monkey studies, and because in WashU I work on the imaging agent for the brain disorders, so for the AD and PD and multiple sclerosis. So that's why monkey study is more relevant to the human brain disorders. So if the in vivo from rodent study cell level on rodent study is more probably move to higher species in monkey studies. If the preclinical study is still very promising, then we move the clinical translation through the different pathways. Typically, the 3 pathways called EIND, IND, RERC. So EIND, just for the first human study, it's called exploratory IND, investigational new drug application, or traditional IND, or RERC. So RERC, that means that the drug already injected to human, the dosimetry toxicity already demonstrated, then move to the second different indications. If then the EIND or RDRC also can be transferred to IND, traditional IND, then go to the subject to the phase 1 or 2, 3 until FDA approval. So I have involved in the whole process and moved to several tracers, pharmaceutical to the first human studies. So this picture shows that FDA approved radiopharmaceutical. We can see that majority are F18 labeled because F18 have favorable half-life, 2 hours, and most of tracer for the brain disorders and cancers. So the 2 hot topic is the tumor, cancer, and the neurodegenerative disorders. You can see the Alzheimer's disease, prostate cancer. That's the 2 major category for diagnosis and for the tumor and the brain disorders. The red one I highlight that the FDA approved, that's a most common use is glucose, because even no matter normal cell or tumor cell or brain disorder, they have the different consumption of glucose. So that's the most often used FDA approved radiopharmaceutical. So this picture shows our infrastructure in our hospital site. We have multiple labs in the DuPont experience station, also in the hospital site. So the first one is a 3T, PET MRI for the clinical practice, or also for the patient survey, that's for the human studies, that located in the hospital site. In the middle, the 2 key piece equipment in the DuPont experience station. That is, one is called G4 PET X-ray, another one is gamma counter for detect the whole body mice to look at the mice. The right side count radioactivity. To when we do the biodistribution study, we dissect the mice, collect each organ tissue to count radioactivity. Then the bottom is looking in the hospital side, that's GMP room. Also, we call it clean room. In clean environment, to the left, the bottom left is the clean room to reproduce radiopharmaceutical in the clean room. The right side to the quality control or radiopharmaceuticals. So we are acting purchasing a small animal 3T PET MRI. So just replace the G4 PET because there's the middle one PET, there is no CT or no MRI function. So we're trying to upgrade this system. So today, the main topic, we are trying to develop the imaging agent for the look at the brain lymphatic system. So here's some background information. So we know that the human lymphatic system play an important role in maintaining the fluid levels and also play a key role in removing the cellular waste and also play a key role in activation immune response, reacting to bacteria or dealing with cancer cells. And also the human peripheral lymphatic system can absorb some of the fat we uptake in our intestine. Then we come out a question, do we have the lymphatic system in the brain? Because for over 200 years, many people thought that there's no lymphatic system in the human brain. So for a long time, the central nervous system, many people thought there's a lack of classically defined lymphatic system. And however, we know that the brain consists of 2% of body weight and consume about 20 to 25% of energy. There must be a way for the brain to remove waste. How the brain remove the waste, that's a debate. There's a lot of studies because we know unknown. So until most recently, two papers, one in the 2012, they first report the lymphatic system. The second paper appeared, the two concurrent paper appeared in 2015. They report the first report regarding the meningeal lymphatic system. So this is the first pilot study regarding the discovery of the brain lymphatic system. The key takeaway is they use optical imaging. They just inject the fluorescent dye to the brain to look at the flow rate of the lymphatic vessels. The key message is we can see that there are some biomarkers there in the body. We get one biomarker is called the VEGF visual endothelial growth factor 3, called VEGF3, the biomarker highly expressed in the meningeal lymphatic system. Then more and more studies, they found that when delete of the biomarker VEGF3 will have the significant impact, will disrupt the growth of meningeal lymphatic vessels. Then the paper, they use a fluorescent dye to look at the vessel current. That's the first pilot study. Then the second study published in the same year, they also do the local injection. The right top, you can see that they use a PEG infrared dye to the animal study dye to inject the animal to track the flow of lymphatic system. In the right in the middle, they just do the two animal models. The transgenic, they just something like just do ligation to look out the lymph node. And the right type, with the right type, the right, we can see that when we inject dye, the red, we can see clearly the dye flow to the lymph node. But to the transgenic from the left side, when there's no lymph node, you can see the waste current was blocked. There's no dye flow to the lymph node because there's no lymph node. We can compare that from the fluorescence. From the right picture, we can see that, and with the normal lymph node, we can see the dye flows through the brain lymphatic system. They go to the lymph node. However, the transgenic mice, when the lymph node with the ligate or lock out, there's very minimal uptake. And also in the body, they do a lot of biomarker analysis. They further confirm that the different biomarker, the unit expressed in the meningeal lymphatic system, and the VEGFR3 is one of the key biomarkers. So seeing the report of the lymphatic system around 10 years ago, there are more and more studies. They found that dysfunction of the lymphatic system and meningeal lymphatic system and relate to many neurological disorders regarding waste current, neurological disorders. They're also associated with multiple sclerosis. They already have animal study demonstrate that they have associated with multiple sclerosis, epilepsy, and traumatic brain injury, etc., many disorders. However, there's lack currently the lack of imaging modalities tracer for non-invasive or imaging of lymphatic system. I showed that majority of study they use fluorescence dye that method. So what we are trying to do is we try to use a PET image tracer label F18 to look at the lymphatic system. The first step, we try to look at the brain, the whole brain lymphatic system by targeting biomarker called VEGFR3. And because there are also a lot of debate, just because one paper they are talking about the lymphatic system discovered in 2012, the meningeal lymphatic discovered in 2015. What's the connection between the system? There's a lot of debate because what's the contribution for each system? What the debate? What contribution for the lymphatic and meningeal lymphatic system? And what's the role in the normal and the neurologic disorders? Because there's some paper before that even our normal brain, when we take a rest, when we have the rest status, when it's during the sleep or during a wake, maybe the weight carries the speed different. Even when the posture, let's say when they stand up, when we lay down, there also some report that the weight carries, the brain stat also different. So there's a lot of unanswered questions. So also we try to clean the disruption lymphatic into the brain, pediatric brain disorder, typically let's say the epilepsy, the multiple sclerosis. They even have some hypothesis that the disruption of the meningeal lymphatic is associated with ADHD. That also there's some paper discuss the topic. So what's the current research gap? So just I show you the pilot study. The research gap, the major gap is the current study, they use optical imaging to look at the brain lymphatic system. The early study, they always use a dye different absorption wavelengths, dye with different absorption waves to look at the brain lymphatic system. The major disadvantage is very invasive. So we can see that they need to inject the dye to the mice, need to sacrifice the mice. That's invasive. There's a study later. There's a study to live animal imaging. However, the disadvantage of live animal imaging regarding because the shallow, the low penetration of dye. So in that case, when we do the live animal study, they need to shave the skull. Means that they need to remove shave the skull, make the skull very thin to look at the live brain. Because when you look at whole brain, the skull is thick. The penetration of the optical imaging very low. You have a challenge to look at whole brain optical imaging. That's a major disadvantage of optical imaging. The MRI based imaging, sometimes the major challenge is they use gadolinium based contrast agent. Gadolinium based contrast agent most probably imaging very often used in the clinical for the patient to look at the brain disorder or others. But the major disadvantage is the gadolinium-based contrast agents, most typically they cannot cross the blood-brain barrier. In that case, we need to do the intrathecal injection. That means that we need to inject drug through the spinal cord. Some study they inject it to the IV injection. IV inject the gadolinium-based contrast agent. In that case, because most gadolinium-based contrast agent cannot cross the blood-brain barrier, in that case, the very small amount can be deposited in the brain and can do smooth imaging. That's a major disadvantage for the MRI-based contrast agent. So our hypothesis is try to develop PET imaging agent, because just as I mentioned that there's a lot of studies show that VEGFR3 first, that the key biomarker, also disrupted. They use a different transgenic mice to look out the VEGFR3, then the development of meningeal lymphatic vessels is disrupted and waste carrier disrupted. So can we just label a biomarker which is F18, try to track the lymphatic growth status in the hairs and the disease? So that's our major hypothesis. So we propose 3 ends, which end have different focus. The first end we mainly focus on the design. We collaborate with University Kansas, design the different compound. Then we try to conjugate compound with radioisotope F18. In the second, when we just as I mentioned in the cartoon, when we design the new compound. So what's the binding affinity? Any change in the binding affinity? Any change in the selectivity? any change in the in vitro behavior, let's say stability. So in the N2, we try to characterize the probes in vitro in the cell level or in the enzyme level, make sure that after we modify the structure, the binding affinity and selectivity keep the same. So N3, we try to evaluate trace in vivo study to look at the in vivo behavior to move from the in vitro cell and enzyme level to in vivo animal level to look at the dynamic PET imaging and the biohistory study and stacked PET imaging. So we also lay out the benchmark. But for this pilot supported by INBRE, we mainly focus on the N1 and 2. So we are actively working on 3. So hopefully we can collect more data to submit R1. So future translation, just I mentioned, we try to, let's say, hopefully we can use a tracer to look at the brain, whole brain lymphatic system. So this cartoon shows a more maybe a clear picture what's talking about the lymphatic system in the brain or meningeal lymphatic system. So that's the whole system, the lymphatic system and meningeal lymphatic called the lymphatic system. So we try to answer the question is, what's the connection between the lymphatic system and meningeal lymphatic system? What's the connection between the brain lymphatic system with the peripheral lymphatic system? Because they're with clearance, they're debatable. Because some people say with clearance through the peripheral lymphatic system, but some studies show that maybe the with clearance maybe through the eye, even through the nose. When the human body sweat through the face, there are also some with clearance. That's a different theory or very different discussions, and we try to figure out what's the connection between the system and what's the connection between the peripheral lymphatic system and what's the contribution for each system in the waste clearance, because maybe the lymphatic system play a key role, maybe meningeal lymphatic system play, let's say, the 50% or 60% role, and also what's the contribution to the for the brain lymphatic to the neurological disorders. Particularly, there's a lot of paper show that particularly the meningeal lymphatic system play a key role in the modulate the immune response in the brain, because for a long time there's a lack of this kind of study, the meningeal lymphatic associated with multiple sclerosis or other immune disorders. That's another key point in the hot topic. They have a lot of publications. So our general research strategy just to show you the right picture. We just use design some compound, do some docking, and do some optimization, region labeling. And if the in vivo behavior is not promising or show, let's say, unstable or show disappointing result, then we go back and collaborate with our collaborators to do some new kind of ligand design to try to repeat the procedure to get a compound with the best in vivo imaging properties. So this we have collaborated with University of Kansas. Just like say our compound with a little compound. This compound have the long high binding affinity with just 3, the 23 nanomolar. So according to the reference, they have the we can modify the different sites, different we color different region, the potential for F18 incorporation. But in one patent, they mentioned that the green one, the right side green one, when we modify the R4 function group, the region 4, have a minimal impact on the binding affinity. That's a key aspect, because they just look at the different site with binding pocket with just 3, the binding protein. They can identify which is the critical point. We cannot change which one we can modify, but with minimal or without affect the binding affinity of a pairing compound. So we try to modify the focus on green part. So that's some chemistry work. There's a long chemistry work. We synthesize the final compound, several final compound. So we collect all the data from University of Delaware. So from the protein MR, C30, MR, F19, and MR and LC mass, we collect data from University Delaware. Then we do some binding assay for the newly synthesized compound. You can see that the top is the target compound. We can say compared to the parent compound, the major changes. You can see the red one is F19, the different linkers, as I mentioned. So we test the binding affinity for VEGF3, and at the same time, we test the selectivity for VEGFR1 and 2. The red box, and all the compounds have very good binding, so in nanomole scale, sometimes 100 nanomole, but the red box, this one have good binding around 20 nanomole, and also the good selectivity for the VEGF2 is 10-fold, VEGF1 is around 30-fold. So after we get the synthesize the target compound, the next step we try to, just as mentioned, we identify the good compound, retain the binding affinity, then we try to do the radio labeling, try to incorporate, because right now the red part is F19, the natural, non-radioactive F19. So we try to develop synthesize radio labeling precursor, also some chemistry work, and the reaction with a two-stage reaction with a reaction in the methylate to incorporate F18. So we confirm by APRC analysis. The top one is the standard. That means that F19 compound at the bottom is the radioactive channel, have the same retain time. And this compound further confirmed by the ALC mass to confirm that the red compound. So we submit abstract. We submit this one to the SNMMI meeting. So we are going to present this result in the next month in the SNMMI meeting. So here, just as I mentioned, this support by INBRE pilot, we mainly focus on A1, 2. So next step, we are our in the MOS, the IHC core immunostaining, Hazel Hardy, the active working biomarker analysis. We try to look at the VGL3 in the brain. So we try to collect more data. I mean, look at the VGL3 in the healthy mice. In later on, maybe we look at the biomarker compared to the disease model to see the biomarker changes. And also we are actively working the radio labeling. We get some preliminary result, just as I mentioned, get some preliminary result regarding radio labeling. So we try to scale up to labeling to also automation of the radio labeling in the clean room, just as we scale up and do the high dose to make sure the precision reliable or reproducible. And also after get the radio labeling done, we try to move to the in vivo imaging studies. So that's the plan. So and look at the imaging to look at the whole brain kinetics of the biomarkers. So hopefully we can submit the R1 in the October. So we try to respond to the PR 25036. This part, the RFA is very, I think, very fit our application. They try to develop PET tracer and SPECT tracer for the biomarker for the central nervous system. So that's the current update regarding the INBRE pilot support project. So lastly, I'd like to appreciate all the collaborators and mentors, and also the funding source from the Department of Radiology and the MOS. And just as I said, all the chemistry data, and I collected all the chemistry data from the University of Delaware. I like to appreciate Dr. Wenji Xu, and he, the organic chemist, he did all the chemistry synthesis work. And recently he found a faculty position, so in China. But he helped the submit abstract. And we have a new postdoc, Hani, and he did all the binding assay. I appreciate all their contribution. So I also have, during the research, I also have several multiple summer research students also make some contribution to the research. And the research, this currently proposed, supported by the INBRE project, partially supported by the NIH grant and the MOS Foundation. The right picture shows the MOS, the radiology research week we take last week. Thank you. That's all my talk. Thank you.